Okay, everybody, welcome. My name is Kieran Samwick. I'm from the University of California, Riverside. Thank you all for coming today. We have a really great lineup of talks on understanding the causes and consequences of recombination rate variation today. So I co-organized this symposium with my colleague Susan Johnston here from University of Edinburgh. So first up, we have Matthew Hahn, who will be talking about estimating recombination using only the allele frequency spectrum.
All right, awesome. I'm a minute early. Should I still start?
Yeah.
Okay, fill time. Jokes. Right, so this project started when I read this paper by a former PhD student of mine, actually Jeff Adrian, when he was a postdoc with Andy Kern. They made a machine learning model called relearn to predict the recombination rate. And it works really well. It's quite amazing, actually. And the standard version of relearn, you basically give it a VCF, and it predicts the recombination rate in your population. And it does it, as I said, really well. The more interesting part for our purposes today is that there's a second half of relearn that does the same exact job, but with pool-seq data. Okay, so that's a little bit stranger. So now you only hand relearn allele frequencies, and then it also tries to estimate the recombination rate. And as I will explain, I'll explain why it's surprising. That's a little bit weird, and this whole project is me trying to figure out what's in the black box of their machine learning method, or maybe it's actually just cheating and it's not working the way we think. And so that's what I'm going to talk about today. So I'm going to tell you why it's surprising that we can do this from pool-seq, but first I have to tell you that when I say recombination today, I only mean the population recombination parameter rho, which is equal to 4 times the effective population size times the meiotic recombination rate. And rho is proportional to the number of recombination events in your sample from nature, okay? So we're always estimating rho, just remember that. People who want to go out and estimate rho have done it in lots of different ways. The most common way probably is to use gametic LD, gametic linkage disequilibrium. So you have haplotypes from a bunch of individuals. You calculate some summary of the association between the alleles on those haplotypes, like R squared, and then we can use classic results or composite likelihood to estimate rho. Right, so you have phase data, you estimate rho from the phase data. If you don't have phase data, if you just have genotype data, you can still estimate recombination rates, but using genotypic linkage disequilibrium. So now you're looking for an association between a genotype, say A1, A2, and another genotype, say B1, B1. Okay, so that still actually has the same exact expectation as gametic LD if you just didn't know gamete phase. There's a slightly rarer way to do this also with a form of disequilibrium called zygosity disequilibrium. So now I only have whether you're a homozygote or a heterozygote. I don't know which kind of homozygote or which kind of heterozygote you are, but SMC-based methods can actually still estimate the recombination rate using that information. The resolution of this method is not very good, but it still tells you the recombination rate. Okay, so the thing that's in common between all those disequilibrium methods is that they require that you've sampled a single individual and that you know its phase or genotype or zygosity, right? And each individual has to have those assigned to them. Pool-seq is quite different because we don't have any labels, right? We've taken all of our, in this case, flies, we've smashed them together, we've put them into two with no barcodes, and we just have a pileup of reads which then tell us the allele frequencies at all of our SNPs. And there doesn't seem to be any of these things that are possible if you just have that pool-seq data. Some smart people have been able to detect very, very short gametic LD, like the length of a read, because in the pool you might still sometimes see a SNP at one end of a read and a SNP at another end of a read and kind of be able to figure out gametic phase. But that's at very, very short distances. And relearn works over very much longer distances. Okay, so that's not what relearn is doing. There's also no read information input into relearn. Right, so what exactly is going on in here to do recombination rate estimation? One possibility, which I thought it might have been doing, is to look at actually levels of diversity. So here it's Drosophila data again. The red line is divergence between two species. The blue line is polymorphism, and the green line is the recombination rate. And because of link selection, there's actually a pretty near universal relationship between levels of diversity and the recombination rate. That's not what they're doing. Their method works in totally neutral simulations. So that's not it. A few clues as I was trying to puzzle this together. So if you just look at the left side, So if you have a pool of n equals 20, so 20, let's just say haploid Drosophila smashed in a tube, and you increase the read depth, your accuracy gets better. So increasing the read depth makes your allele frequency estimates better. And if you jump from n equals 20 smashed into a tube to n equals 50, you also get more accurate. And that also increases the accuracy of your allele frequency estimates. So that suggests that there's something about the allele frequencies themselves that are telling relearn about recombination. And so the allele frequency spectrum is this classic thing that we think about in population genetics. We know exactly how it should look, especially in a neutral equilibrium population. There's lots of singletons, and then they kind of go down in frequency as you get higher and higher. The problem is that it's the same allele frequency spectrum in every equilibrium population, no matter what the size is. It does change with changing population size, but again, relearn works even in neutral cases across lots of different recombination rates. So at this point, I'd like you all to try to take a guess as to what relearn is doing. Internally is fine. And you can also guess that it's cheating, right? I mean, lots of machine learning works because there's some sort of data leakage or something else. But okay, now I'm going to tell you how it does work, and it actually does work. So it is the allele frequency spectrum, but the trick is that you have to think about the allele frequency spectrum at every marginal tree, right? So here in my toy example is a tree at one locus on the chromosome, and I've thrown down some mutations. So there's 3 mutations. 2 of them are on a tip branch. We call those size 1 because they have 1 descendant. And then there's 1 on an internal branch with 2 descendants, so it's of size 2. And so my allele frequency spectrum in this very simple example would look like that, right? I'm going to assume infinite sites, so there can't be the same mutation twice here. If I think of another marginal tree somewhere else on a chromosome, it might look like this, and I can throw down my fake mutations. And again, I have 2 mutations of size 1 and another mutation of size 3 up here. And I just chose not to put any of size 2. So I get a different allele frequency spectrum. And it's not just because I chose slightly different mutations, but because if you look at this, this tree actually has no way to get size 3 mutations, right? So every tree has a marginal allele frequency spectrum it can kind of emit, but no tree can emit the true allele frequency spectrum. Here's just a more complicated tree. So I can always do size 1 on the tips, size 2 here, size 3 on this long branch. There's other mutations of size 3 that can appear there, mutations of size 6 that can appear in the ancestor of these 6, and mutations of size 7. So if I imagine a possible allele frequency spectrum, it looks like this. But there's no way to get mutations with a single mutation of size 4, size 5, size 8, or size 9 on this tree. If we think now about how the trees change along a chromosome, I can start with one tree, and then I get to a recombination event. The recombination event changes the tree a little. I go again, I wait till another recombination event, it changes the tree a little. And I can imagine that I have a different emitted allele frequency spectrum from every tree along the way. And it'll change a little bit. Sometimes, all the trees can have mutations of size 2 or size 3, but the length of those branches that subtend 2 descendants are longer or shorter, so there will be more or fewer mutations in this category, for instance, size 2. So what I think relearn is doing is basically it's looking at a low recombination region, and it sees, effectively, it doesn't never see the trees, but it sees a allele frequency spectrum that comes from only a very few trees. And if you're in a high recombination region, your allele frequency spectrum is the sum of allele frequency spectra across all of these additional marginal trees. And as you add more and more marginal trees, you actually approach the equilibrium expectation, which is actually the expectation across infinite numbers of trees. Right, I said this earlier, but actually along the way we had to prove to ourselves that there's no single tree that can give you the expected spectrum. In order to even get all of the size mutations, you need a perfectly asymmetrical tree. But under the coalescent, the asymmetrical trees you get will not give you this expected spectrum. So it requires many, many trees to get the expected spectrum. So we built our own version of relearn where we could control exactly what was going on. And as a little baby version, we make sure there's nothing leaking in, there's no extra data sneaking in the back door. We did all of our simulations in MS Prime. It's a 50 KB window with different levels of recombination. The MS Prime produces an allele frequency spectrum. We then normalize it, so now our machine doesn't get any information about even how many SNPs there are. It's just a normalized vector of allele frequencies. It has no idea where they are in this 50 KB window. or how many there are exactly. Then we trained our simple machine, which we call NoDear, no disequilibrium estimator of accurate recombination. And underneath the hood of NoDear is XGBoost, a very common machine learning model, which works great with vectors. And the training we do is simply a labeled data set where we say, here's a simulated data set with this value of rho, and here's what the allele frequency spectrum looks like. And here's another much lower value of rho, and here's what the allele frequency spectrum looks like. And we just tell NoDear, learn these relationships, and then I'm going to give you another vector. Please predict what rho is. First, we looked at the data, and actually it looks like what it should look like. I'm showing you the folded spectrum here, so I made it a little bit harder on myself. I didn't even know what was derived or ancestral, just the minor allele. In low recombination, it's very jumpy. In high recombination, it's quite smooth and looks just like you would expect. And this is actually how it does. So there's something like 7,000 data points here. At very high recombination rates, it can't really tell the difference. There's a lot of trees. Everything looks like the expected value. At very low recombination rates, every window has 1 or 2 trees, and it can't really distinguish among them. But in biologically realistic regions of parameter space, NoDear is really, really good at predicting what your recombination rate is just from seeing your vector of allele frequencies. If you think that still seems like magic, I think you could all actually do this task if I rephrase it as, I tell you there's a normal distribution, and I show you the histogram from my samples. You could tell me that, oh, you probably don't have that many samples there, and you have lots of samples there. And in this case, our samples are the numbers of trees, because the number of trees in a window is the number of recombination events plus 1. Okay, so the number of trees is our number of samples, and we can tell when we have lots of samples or few samples. So just some considerations about real data. How big a window should we use? So I was using 50 kb windows in the results I showed you before. If we make the window bigger, the regions on the end get a little bit fatter, especially up in high recombination and down in low recombination, but we still have pretty good power. If we make the window much shorter, we lose a lot of power. Most of this is because we don't have enough SNPs, right? So you need to be able to tell what the allele frequency spectrum is. And as your window gets shorter and shorter, it's not necessarily that you have few trees, but that you have not enough SNPs. How does it compare to the state of the art? So you could use NoDear. Actually, I recommend that you use Relearn, not our toy. Or you could use a very good composite likelihood model. So here's another set of simulated data. Here's NoDear. It predicts rho really pretty well, I would say. Here's Pyro, the latest and greatest. It does really well, but like it's fancy, and this is logistic regression with some bells. So I think we're doing really well. The other advantage is this is still with 20 individuals in my pool. If I increase it to 100 individuals, I get a better estimate of the allele frequency spectrum, and my accuracy gets better. I can also, this is all done in equilibrium populations. I can also do it in a non-equilibrium population, and NoDear seems quite robust to that. So now my model is trained in an equilibrium population that's predicting in a non-equilibrium. And it seems like it's actually predicting that choppiness and not exactly the shape of the allele frequency spectrum, which is actually a really nice thing. And we do essentially equivalently to Pyro under these conditions. How about real data? We don't know the real answer, but we took 1,000 genomes data from Finland. We removed all the genotype information, just made it an allele frequency spectrum, and we get a map of recombination that actually is highly correlated with the Pyro map for the same population. Okay, so in conclusion, we can learn the recombination rate with just the allele frequency spectrum, and it's because we can consider all of these marginal trees as our samples. If you really want to do this in pool-seq data, it's worth it. Relearn is extremely accurate. You should just use Relearn. But I was interested in why it worked. And then the second thing is that machine learning is becoming more common. And we think of it as this black box, and sometimes that's a bad thing, and sometimes actually these methods tell us something about the data that we might not have noticed before. So with that, I'll thank Sarthak, who's a grad student in my lab, who helped with all this. And I will declare here that we will submit this to genetics. Hopefully you see it there eventually, and because it's a really good journal. Thanks. We have not, but I assume it's not so different from non-equilibrium, we've tried structured populations, we've tried gene conversion, it never really gets much worse. And because, those all do change the equilibrium spectrum, but they don't change the choppiness among all of those bins. Mark?
Can you get anything out of the autocorrelation?
Yes. So, absolutely. So what I didn't say is that relearn is probably doing a little bit more than I'm giving it credit for, because it does have the location of all the SNPs in the window. So you could absolutely look at how the allele frequency spectrum is correlated along the chromosome and do more. Yeah.
Okay, thanks very much, Matt. Okay, our next talk is by Carol Veller, who's going to look at the heritability of recombination.
Thanks, Susan. And this is a question that I've been thinking through with Susan Johnston on what estimates of the heritability of recombination mean. So recombination happens in meiosis. At meiosis 1, the chromosomes replicate. Homologs look for each other in the nucleus via a process of double-strand breaks and homology search. Once they've found each other, a subset of those double-strand breaks mature as crossovers, which physically tether the homologs together before they segregate in the first meiotic division. There's then a second meiotic division to produce gametes. And maternal and paternal alleles are shuffled in those gametes. They're recombined. This recombination happens along chromosomes because of crossovers, and it happens between chromosomes because of independent assortment of chromosomes, Mendel's second law. And recombination is important for a lot of reasons. It serves a structural function in meiosis 1 with crossovers tethering homologs. This is required by most, though not all, species for proper segregation of the homologs. But evolutionary biologists tend to be more interested in the genetic consequences of recombination, that it shuffles allelic combinations. This can make adaptation within a species more efficient, but it can also break apart co-adapted combinations of alleles. And these kinds of considerations have been at the heart of a very deep, very rich literature in evolutionary biology that has asked questions like, should a species recombine at all? That is, should it be sexual or asexual? If it is sexual and does recombine, how much should it recombine? It's often been claimed that the amount of recombination is under stabilizing selection within a species, and that species differences in the amount of recombination might reflect adaptation. There's actually surprisingly little empirical evidence for those two claims. I've highlighted an exception here from one of our symposium organizers. But any claim of adaptation of the amount of recombination requires that it be a heritable phenotype. And I'm going to consider both of those words. So what is the phenotype? And once we've decided what the phenotype is, what is its heritability? Now, the easiest way to measure the amount of recombination is just to count the number of crossovers in meiosis, or alternatively, the number of crossovers in gametes. That's conceptually the simplest measure of the amount of recombination. It's the one that's most often reported. It's related to important metrics like genetic map length. It doesn't take into account the role of independent assortment of chromosomes in recombination. A metric that does is Darlington's recombination index, which is the number of crossovers plus the number of chromosomes in the haploid set. This is, corresponds to the number of freely recombining chromosomal segments in meiosis. It in turn doesn't take into account the importance of the positioning of crossovers on chromosomes for the amount of recombination that they cause. And so perhaps the most direct metric for the amount of recombination is the proportion of locus pairs or pairs of genes in the genome that recombine each meiosis. But for the purpose of this talk, I'm going to take the simplest possible measure, the number of crossovers, with the expectation that what I say will carry over to these more nuanced metrics as well. Okay, so that's our phenotype. What does variation in that phenotype look like? These are numbers of crossovers in meiosis 1 nuclei for a bunch of species, measured cytologically using immunostained spreads of chromosomes like displayed at the top right there. And you'll immediately notice that there's enormous variation across nuclei in the number of crossovers that they contain. A natural question to ask is whether this variation is coming from intra-individual variation, variation across the nuclei produced by the same individual, or systematic differences between individuals in the number of crossovers. And to address that, I'll focus on the case of human male. These data come from 10 individuals, and if I plot them individual by individual, this is the picture that you get. Of course, we're not really interested in crossovers per myocyte we're really interested in crossovers per gamete, and so I can artificially simulate gametes from these myocytes to obtain the distribution of crossover number per gamete for different individuals. And you can see that almost all of the variation here in crossover number per gamete is driven by intra-individual variation in that phenotype. In fact, of the total variation in crossover number per gamete, only about 1/20th derives from systematic differences between individuals in their average crossover numbers. Okay, so with this enormous intra-individual variation in mind, let's return to the question of what the phenotype is that we're interested in the heritability of. Okay, so here's a population of individuals, each producing many, many myocytes, resulting in many, many gametes. One phenotype we could be interested in is the average number of crossovers among the gametes produced by an individual. Right? That's a phenotype we can measure, we can estimate its heritability. I would argue that an evolutionarily more relevant phenotype is the number of crossovers in a randomly selected gamete from each individual. This is maybe easiest to appreciate in a low fecundity species like ours, where each individual is having 1, 2, 3 offspring. And each of those, and if selection is acting on the genetic consequences of recombination in those offspring and their descendants, then selection is only seeing the number of crossovers in the gametes that produce those offspring, so 1, 2, or 3 gametes from each individual. So these are the two phenotypes I'm going to consider the heritability of: the average number of crossovers among the gametes produced by an individual, and the number of crossovers in a randomly selected gamete from each individual. So let's take the first one. Okay. The number of crossovers among gametes is just 1/2. The average number of crossovers amongst gametes is just 1/2 the average number of crossovers in meiosis I myocytes, which will be a useful metric to start with. So their heritabilities will be equal. I'll designate this by m bar. Its heritability is its additive genetic variance divided by its phenotypic variance, and it has an upper bound of 1, of course. Okay, so before we consider the heritability of crossover number in a random gamete, let me consider an intermediate phenotype, a randomly selected myocyte. The number of crossovers in this myocyte is the mean for the individual plus a noise term that comes from the fact that I'm sampling a random myocyte. And so the heritability of the crossover number in a random myocyte is its additive genetic variance divided by its phenotypic variance across the population. And if you focus on its additive genetic variance, you'll notice that this is driven entirely by additive genetic variance in the mean number of crossovers across myocytes. The random noise term from sampling is, of course, not heritable. Technically, it's linearly independent of individuals' genotypes. And so I can replace that with the additive variance of the mean number of crossovers, and I get something that looks like the heritability of the mean number of crossovers. I can rearrange terms, and indeed it is the heritability of the mean number of crossovers multiplied by this dilution factor. The dilution factor is smaller if intra-individual variation in crossover number across myocytes dominates the overall variation. Okay, so now let's look at a randomly selected gamete. It descends from a randomly selected myocyte, and so is subject to the same variance from choosing a random myocyte. But the number of crossovers that it inherits from that myocyte is also random. It's binomially distributed according to Mendelian segregation. And so we actually have two kinds of intra-individual noise among gametes. We have noise from the fact that there's variation across myocytes in the number of crossovers, and there's noise from the sampling of crossovers from myocytes to gametes. And we can characterize that second variance addition to the variance mathematically. So if we now look at the heritability of crossover number per gamete, which I think is the evolutionarily relevant heritability, right, the additive genetic variance there again is driven by the additive genetic variance of mean crossover number. And so we're going to again get something that looks like the heritability of mean crossover number, but with a dilution factor, which is smaller than the previous dilution factor, being driven now by variation across myocytes within an individual and their number of crossovers, and the additional variation from Mendelian segregation of crossovers from myocytes to gametes. Okay, so here we have three expressions for the heritability of different phenotypes. The parameters in these dilution factors can in principle just be measured from data very easily. So let's do that again in our case of human male. These are our three phenotypes. Let's set the heritability initially of the mean number of crossovers to 1, its upper bound. This will allow me to derive upper bounds for the heritabilities of the number of crossovers in a randomly selected myocyte and a randomly selected gamete. The upper bound for the heritability of crossover number in a random myocyte is 22% in human male, and for a randomly selected gamete it's only 8%. If I now set the heritability of the mean crossover number to a more reasonable quantity like 30%, that's actually quite well to the high side of estimates that have been obtained, I now find that the heritability of crossover number in a randomly selected gamete is only 2%. Okay, and I want to stress that that is, amongst the kinds of phenotypes that we usually consider a really, really small heritability. It's going to substantially impede any adaptive response of the crossover number to selection, and in sufficiently small populations, it will actually prohibit fine-scale adaptive tuning of the number of crossovers. And again, this is getting driven entirely by enormous intra-individual variation in the phenotype. Okay, so I'll leave you with a few questions. First, from a kind of physical and molecular perspective, what drives this enormous intra-individual variation in crossover number? This is something that we know something about. It's driven almost entirely by a phenomenon where the number of crossovers within individual nuclei co-varies positively across the chromosomes. So if I take a nucleus from an individual and I look at chromosome 1 and it contains an unusually large number of crossovers, then so typically will chromosome 2 and 3 and so on in that nucleus. That phenomenon of crossover covariation is in turn driven by an analogous per-nucleus covariation across chromosomes in how their chromatin is packaged at the relevant time step of meiotic prophase. And the factors that are governing that global regulation of chromatin packing are a very active area of study in the molecular biology of meiosis at the moment. A second question: why does selection tolerate this enormous intra-individual variation in crossover number? As I've argued, it substantially reduces the heritability and therefore impedes the response to selection. If crossover number is understabilizing selection within populations, as it's very often claimed to be, then this variation is intrinsically deleterious. So why is it tolerated? Well, maybe selection on crossover number is very weak. Provocatively, maybe there's no selection on crossover number except subject to certain physical constraints. Perhaps there are adaptive explanations for this variation. And finally, these problems apply specifically to the phenotype of crossover number in a randomly selected gamete. I've argued that that's probably in many instances the most important phenotype here. But are there scenarios where selection doesn't act necessarily on that phenotype, but instead on an individual's mean crossover number across all the gametes that they produce? Or alternatively, perhaps selection in some scenarios acts on an individual's distribution of crossover number. And with that, I think I'm finished quite early. I'll just plug the fact that I recently started a lab at the University of Chicago. The lab's going to work on predominantly theoretical questions in evolutionary genetics. We're hiring at all levels. So if you're interested in these kinds of topics, then and you wish to join the lab, please get in touch. Thank you.
Are there any questions?
Always, Mark.
Will the response to selection in recombination rate be determined by the heritability or by the aggregate genetic variance?
Let me think about that. I mean, the heritability, I would say, but we can talk about that.
So I apologize if I missed this, but it seemed to me that the intra-individual variation in recombination rate, yeah, you're treating it, whether it's Gaussian or however you're looking at the distribution of it, but in fact we know in chromosomes that it's structured, right? In certain parts of the chromosome, it's higher or lower, whatever, and so in fact you have to model that quite differently. So you're treating kind of your bowl, here's one bowl of oranges of different sizes for one individual, another bowl of oranges, but you've got an orange, an apple, whatever, combination. And that's again sort of thinking about that question. It's going to affect in particular, your denominator a lot.
Yeah. Yeah. So what precisely the correct metric is to think of in terms of recombination's effect is obviously a big question. I focused on the metric that pretty much everyone uses and everyone measures the heritability of, but I am very interested, and I've thought quite a lot about what metrics that capture the functional effect of recombination, and those might be subject to lower variance. You're right.
Yeah, I was going to say, I would be very
recombination rate. And
Yeah.
so maybe it's all driven by segregation areas subsequent to recombination.
You mean like the heritability of propensity to aneuploidy? Is that what you're? I mean, I expect that is quite heritable, but it's.
Okay, thanks very much, Carl. Our next speaker is Tom Parée, who will talk about selection for recombination.
Hello, everybody. Thank you for giving me this opportunity. So today I'm going to present you why selection for recombination is uncoupled from adaptation. So we know that there is a variation in recombination rates, for example, between species. Famously, there is more recombination in the fish and yeast as there is in dogs, for example, because more or less crossing over. Importantly, the position of this crossover is not uniform along the chromosome, with typically more crossover arising in the periphery of the chromosome, leading to this heterogeneous recombination landscape. And there is some irritability for recombination with intra-species variation, which is due to recombination modifier gene with allele that encode for more or less crossover and different position of crossover. So a question that must interest a lot of us in this room is whether this recombination rate diversity is adaptive or not, which in population genetics term mostly translate to can a recombination modifier be indirectly selected because it produces beneficial adaptive combination? Why indirectly selected is because it creates some genetic combination at some locus and then it needs to spread by association with this genetic combination. So to a bit explore indirect selection, I've simulated some toy model. So for example, here we have a modifier M that increase recombination and that is found nearby selected allele in red and green. And here we see that the recombination modifier is selected for because it creates some beneficial adaptive combination and can spread by hitchhiking with them in the population. Now, if we have the exact same model, but we put the modifier more distant to the fitness loci, we see that the modifier is not selected, just because there is too much recombination between the modifier and the selected allele, and it cannot maintain its association with the adaptive genotype it produces. So we see here that the position of the modifier matters, and more generally that indirect selection on a modifier is due to its local effect. If it's able to create adaptive genetic combination, it's its local genomic neighborhood. But still, the modifier here has a genome-wide effect, and so all the effect of all the genome-wide adaptive genetic combination create will impact adaptation. So maybe already you understand better why I said that indirect selection on modifier is uncoupled from its genome-wide effect on adaptation, which is genome-wide. Here, I think I have even more interesting with a heterogeneous recombination landscape. So if you have a modifier that increase recombination around him, but it decreases it in a nearby interval. And now we have a second model where exactly the same, but the modifier is the interval where it reduces recombination. And maybe as you anticipated, when the modifier is in the region where it increases recombination, it is favored because it increases this local efficacy of selection. But when it's the interval where it decreases recombination, it's selected against because it decreases the local efficacy of selection. So to study this, I use the REC1 recombination modifier gene in C. elegans. So the wild type C. elegans recombination landscape is classic with more recombination in the periphery than there is in the center. But you see that what does the REC1 mutant is to smooth the recombination landscape, so it tends to equalize recombination along the chromosome without changing the number of crossover. So if we take the difference in recombination rate, we see that in some genetic interval it will increase recombination, and in some genetic interval it decreases recombination. And importantly, the REC1 mutant doesn't seem to have any effect on fertility, problem of aneuploidies, and so no direct selection. So the effect of the mutant is genome-wide. In green, you see the chromosome tree, is what just I showed you the slide before, and you see that on all chromosomes, it increases or decreases recombination in some interval. And what's interesting to see when we look at the SNP diversity in C. elegans, there is a correlation between the inverse correlation between genetic diversity and the effect of the mutant on recombination, with most of the genetic diversity in region where the mutant reduces recombination. So one prediction that we can have from this is that the mutant should impair adaptation by reducing genome-wide efficacy of selection. But now I would like to draw your attention on the chromosome 1. And you see that is a chromosome that bears the REC1 gene. And around its own genomic position, the REC1 mutant increased recombination. So one prediction, because we saw that indirect selection is local, is that the mutant could be favored despite decreased adaptive genome-wide adaptation because it increased the local efficacy of selection. So to test our hypothesis, I constructed a genetically diverse population that come from sex 16 wild isolate and that have been adapted to a lab environment. And importantly, in this population, there is a high outcrossing, so a lot of genetic mixing, a lot of effective recombination. And you can assume for this presentation that we can neglect selfing. And we introgress the REC1 mutant and wild-type allele in this genetically diverse and outcrossing population. So here we have two populations with the fixed wild-type or mutant allele, and you expect that the effect of the mutant is genome-wide, and it will affect the genome-wide adaptation, which is our first question. Does the REC1 mutant will impair genome-wide adaptation by reducing recombination in the most diverse region? But here the REC1 mutant is not allowed to evolve. But when we mix the two populations, here the REC1 mutant can spread or decrease in the population. And here we hypothesize that it should be favored because it increased this local recombination rate. So in the first experiment, where I have a REC1 wild type or mutant population with fixed allele, we look at the fitness after and before 40 generations of adaptation to high salt, which is a stressful environment. And we see that if we look at this, the fitness change, the mutant have a lower fitness increase that we observe in the wild-type population. Each dot in an independent experimental evolution population. And so if we wonder why, I've done a pool sequencing. And I calculate this matrix, which is a correlation between different alleles. So if there is a higher linkage, we accept there is higher correlation in the dynamics of selected allele. And if there is an inversely. So basically, this measure is mostly a linkage. And so it should not surprise you when the region where the mutant decrease recombination, it increase linkage. The region where it decrease recombination, it increase recombination, decrease linkage. But what is the most interesting, if we look at genome-wide, we see that the mutant increase linkage disequilibrium. Because again, most of the genetic diversity is where the mutant decrease recombination. So this genome-wide increase in linkage seems to be able to explain why the mutant decrease genome-wide adaptation. Now, if you look at the evolution of this REC1 mutant within a polymorphic population, so where it's able to evolve, and where there is genetic mixing between the wild type and mutant allele, and so it does not stay associated with most of the genome and most of the genetic combination it creates. So I tested for selection coefficient of the mutant. So we measure the frequency of the mutant, and from that I can infer selection coefficient. And you see that on average, the REC1 mutant is selected for, which is consistent with our hypothesis that the mutant could be favored because it increases recombination rate locally. A good evidence that what we see is indirect selection is that if we increase population size, we reduce the selection on the REC1 mutant, which is consistent with this population genetics prediction that in large populations there is lower selection for recombination due to something that we call the Hill-Robertson effect. So to summarize a bit these two experiments, I've run a simulation that aim to mimic the experimental framework. So it's the same genetic diversity as the experimental population and the same recombination landscape, wild type and mutant. And so we see that when we simulate a wild type and mutant population, with the simulation we recover that the REC1 mutant have lower adaptation, which at least in this simulation can be fully explained because recombination is decreased in most diverse region by the mutant, and so it decreased the genome-wide efficacy of selection. Now when we simulate a population where the REC1 mutant is polymorphic, as the experimental result where you recover that the recombinant mutant is favored despite a decreased genome-wide adaptation. And in this simulation, I can look at the association of the mutant and the adaptive loci. So we hypothesize that indirect selection is due to this association that the mutant creates with the beneficial adaptive combination. And we see that in its local genomic neighborhood, the mutant is able to create association with fitter genotype. And for most of the genome, it doesn't create fitter or less fit genotype because it's too far. If you have something to remember for that, I'm sure it's not the first time you've been exposed to this idea for other reason maybe, but the adaptive consequence of recombination are different than selection for recombination, which is mostly driven for a really tiny part of the genome. To understand how recombination modifier evolve in nature, we need to be able to identify where are these recombination modifier, what are the loci that are under selection, and what is the association between the modifier. And this model can seem a bit complicated in some aspect, but it's already much more simple that we observe in the wild, where you have probably multiple recombination modifier that maybe have some genetic conflict, some local modifier that don't suffer with uncoupling with genome-wide because they don't have genome-wide effect. So just one more slide to show you that it's really unintuitive sometimes, the pattern that we observe on indirect selection. Is that here, you know that C. elegans is sulfur, and what I show you was under main node crossing. But here I simulate that if we increase the selfing rate, we can look at the selection on the REC1 mutant, and we see that for very high selfing rate, the REC1 mutant is selected against. And so it's when there is low selfing rate, it's what I showed you before, selection is mainly local. But under very high selfing rate, there is not a lot of genetic mixing, and so the REC1 mutant begin to be associated with the whole genome. And we saw because genome-wide adaptation is lower in the mutant, it's a genome-wide deleterious effect, explaining that the mutant would be selected against. So very exciting prediction that Laurence from my lab tested, but I was not here. And so we see that in experimental population, we recover this relationship that under low selfing rate, the mutant is selected for, but under very high selfing rate, the mutant is selected against. Thank you.
Questions?
So my understanding is that there's also differences along chromosomes and C. elegans and gene densities.
Yeah.
And that there's higher gene density in the lower central region, and that's where recombination may increase.
Yeah.
And so I was wondering what your thoughts were about that.
So yeah, like I show a really simple explanation with just genetic diversity, but obviously there could be like a difference in irritability of fitness between different regions, notably because there is more gene in the center. But what we show is that there is more irritability for fitness in the periphery of the chromosome, at least in our population. And maybe one of the reasons for that is we know that in the center of C. elegans there is a lot of essential gene that may be not really diverse, because when we mutate them, they're probably, the mutation are probably deleterious and purge. And so there might be not a lot of irritability for fitness, despite there's a lot of gene.
Oh yeah, sure. I was just curious if anything's known about the frequency of REC1 in natural populations.
Yeah, that's a very good question, because we saw that it can be selected, so we can wonder why we don't see it more often in C. elegans, because like in synaptitis, this recombination wild-type landscape is really conserved. I look in the sender, which is a collection of strain, and there is some variant in some of the senders there, but we don't know like how it affects the phenotype. But as we saw, if we increase a lot of selfing, which is the case in experimental population, it should be selected against. Also, here we don't see a direct fitness cost in a lab environment, but maybe with more temperature variation in the wild, it could be selected again just because it fucked up meiosis at some point. Sorry. And yeah, that's it.
Awesome. Yeah, good to see you. Thank you everyone for coming to my talk, and thanks again to the organizers for allowing me to share my work during one of the symposia. I'm really excited to share with you all today a project that I've been working on for the past couple of years, exploring meiotic recombination as a potential sex-specific source of new mutations in Drosophila melanogaster. So I want to start off my talk by first covering, okay, why do we think that meiotic recombination might be mutagenic? And so first off, just from like a theoretical standpoint, most of you guys probably know that meiotic recombination is facilitated by the creation and repair of a bunch of double-strand DNA breaks. And if you study DNA damage, typically we think of double-strand DNA breaks as bad and mutagenic. And there is also some evidence to support this as well. So the first kind of evidence that I want to cover that has been shown in a lot of different species, here specifically it's in humans, is the relationship between nucleotide diversity and recombination rate. So areas of the genome with more recombination tend to have more diversity. However, there are many other more parsimonious explanations for this specific example. So the evidence that I want to focus on a little bit closer comes from, this is an example from Haldorsen et al. 2019. So here, parents and offspring were sequenced, crossover events were identified, and new mutations were identified. And what they found is that the rate of single nucleotide variants was roughly 30 to 50 times higher within 1 kb of a crossover event. So this is like pretty strong correlative evidence that crossover events are associated with new mutations. And so one final piece of evidence that I also want to talk about before I get into like how we tested this comes from this yeast paper. This is Rattray et al. 2015. And they actually found not only that in Saccharomyces cerevisiae, meiosis was associated with a higher mutation rate, but that if you turned off this SPO11 gene, which is responsible for creating those double-strand DNA breaks, that effect went away. So yeast that did meiosis but didn't do recombination no longer had an elevated mutation rate. So we have pretty strong evidence to lead us to think that these double-strand DNA breaks are causing some amount of new mutations or are strongly associated with them. And we can test this in Drosophila. So what did we do? We performed a mutation accumulation study in different recombinational treatments. So what does that mean? First off, if you're not familiar with mutation accumulation studies, they're incredibly powerful to help you measure mutation rates. So the way that they work is you start with a common isogenized ancestral population, and you set up experimental lines, which you then bottleneck repeatedly for many generations. Because you're bottlenecking the populations, you're inbreeding them, right? It reduces the effective population size and also therefore reduces the ability of selection to purge mutations. And so at the end of your experiment, you have all these lines which have diverged and they've accumulated mutations at the rate at which they've spontaneously occurred. So it's an incredibly powerful technique. And Drosophila are such a powerful system to study this in because there's an SPO11 homologue, MEIP22, which is known to be essential for creating those double-strand DNA breaks and facilitating meiotic recombination. And so there's a strain that we obtained that actually has an interrupted version of that gene, and so those flies do not do meiotic recombination, and they still produce viable offspring, which is super cool and allows us to do these experimental evolution projects. So we set up 2 sets of mutation accumulation lines, one set which was performing recombination and one set which wasn't. And keep in mind, once again, when I'm talking about performing recombination, I mean that they're performing the programmed double-strand DNA breaks that are allowing recombination to happen. We weren't using like balancer chromosomes or anything like that. And another quick thing that I want to mention is that most of the double-strand DNA breaks that are caused by MEIP22 don't result in crossover events. So you might be thinking, okay, well, there's maybe like one crossover event, but there's many more actual double-strand breaks that get formed and then have to get repaired. So we think that there's plenty of opportunity for these double-strand breaks to be acting in a mutagenic way. One final thing, and you're like, okay, well, you said sex-specific. Where's that? Drosophila melanogaster, actually the males are achiasmatic, meaning that they don't perform meiotic recombination in their germline. So our logic for this project was as follows. If meiotic recombination is mutagenic and it only happens in the female germline, then by definition, it's a sex-specific source of new mutations. Okay, so we ended up sequencing 35 lines after 30 generations of mutation accumulation. So that took about 15 months just for those bottlenecks. We sequenced them using Illumina NovaSeq X. I mapped our reads using BWA MEM2, and then called variants using GATK's haplotype caller. We did, if any of you guys have analyzed sequence data, you get a lot of false positives. We filtered out using many metrics, but one that is really important for me to mention is that we only called variants that were present in one of our mutation accumulation lines, right? Because our ancestral lines had some variation different from the reference genome. And so to control for that, we only took mutations that were present in one of our lines. Okay, so what do we find? Across all of our lines, we found 1,064 mutation events, which we categorized into 4 groups. The first, I'm sure you're all familiar with, single nucleotide variants, substitutions. We found 678 of those. We found 235 indels, so short insertions and deletions. And then we also called what we're considering multi-nucleotide events. So if within a 1 kb window we found more than one SNP, we called that a multi-nucleotide event. And then if within 1 kb window we found at least one SNP and one indel, at least 2 events with one of them being an indel, we called that a complex event. We haven't so far found anything super interesting with the multi-nucleotide events or the complex events, so I'm not going to reference them anymore throughout the remainder of this talk. Okay, what do we find when we compared groups? We found the opposite of what we expected. Woo! Yeah, so we found that when you compared across the group performing meiotic recombination and the group not performing meiotic recombination, the total number of mutation events was actually elevated in our group not performing recombination. And that's being driven by an effect that we see, a much stronger effect that we see in our indels. So you can see here, the red indel bar is significantly higher than the blue indel bar, and that significance was much stronger than the total red bar being higher than the blue bar. We also have looked at the spectra of single nucleotide variants, right? So these are SNVs. This was not different between our 2 groups, once again indicating that we don't see any differences in single nucleotide changes between our 2 groups. If you're not familiar with Drosophila mutation experiments, the C to T group is normally high, so that was a bit of a sanity check. It aligns with prior Drosophila mutation accumulation studies. And then something that I'm really excited to share with you guys is I actually looked at what I'm considering like the spectra of indels. So I looked at the ratio of insertions to deletions, and I also looked at the average size of the indels that we found. And I did find a couple of interesting, significant differences between my 2 groups. So you'll see here this is a violin plot. It shows like the distribution of the size of the indels. And my meiotic recombination group, the blue one on top, you'll notice that the size of the indels is really more tightly clustered around 0 compared to my group not performing meiotic recombination. So what this showed us is that meiotic recombination is associated with significantly shorter indels. We also found, when comparing the ratio of deletions to insertions, so like how well is that distribution centered over 0 effectively, that meiotic recombination is associated with a higher ratio of deletions to insertions. So you'll see in the group performing recombination, it's really nicely centered around 0. The red distribution, my not recombining group, you'll see that there's like a right tail, right? There's many more insertions than deletions in that group. So there's almost like a bunch of deletions missing. So these are all preliminary results. We haven't finished going through the data set. I just got the sequence data in May, so I've been frantically trying to prep for evolution. But here are some preliminary conclusions that we have based on our analysis so far. So first off, we don't find any significant difference in the single nucleotide variant rate or spectrum with respect to recombination function. We find fewer indels, shorter indels, and a higher proportion of deletions relative to insertions in our recombining lines when compared to our non-recombining lines. And so to speculate a little bit here, this actually might suggest that the MEIP22 gene is not only important for double-strand DNA break formation, but that it also might play a role in the repair of those kinds of breaks, which would be interesting because our data suggests that in Drosophila, while recombination might innately act as a sex-specific source of DNA damage, it might also be associated with a heightened sex-specific DNA repair mechanism, which is really cool. So obviously, we're nowhere close to being done with this data set yet. there are plenty of additional things that we want to look at. First, we want to dig into a little bit more about where our variants are located. So we want to look at, like, are they in genes? Are they not in genes? Introns, exons. We want to look at, like, whether there are silent variants versus nonsense, missense variants. And then we also want to look at, in Drosophila, there aren't recombination hotspots the same way there are in humans, but we know that certain regions of the Drosophila genome have more or less recombination. So we want to look at different windows and see, in regions with higher recombination rates, do we notice anything about where our variants are located? I also want to look at structural variation. We're planning on using DELI for that. And then another thing that I'm super excited to look at is transposable element mobility. We know there's an evolutionary relationship between TEs and recombination, and we have an opportunity to look mechanistically and see if we notice anything going on. And we plan on using the McClintock pipeline for that. So I think I got done a little early, so I want to quickly thank my advisor, Nathaniel Sharp. He's super helpful. Johanna Abraham, who was an undergrad who helped with all of the labor-intensive fly work on this project. And then Denise, who's a research specialist in our lab, she helped with all of the DNA preps for sequencing. And with that, I will take some questions. Thanks, everyone.
Okay, we have lots of time for questions.
Yeah. So we didn't sequence the parents because we did 30 generations of bottlenecking. However, any mutations or variants that were present in the parents, we screened out in our filtering step, if that makes sense. Yeah, good question.
So I'd like to suggest a natural experiment. So there are species where there's male recombination, and it's kind of like weird, non-canonical. So in Drosophila onaceae, they gained recombination crossing over in the male germline. And so, I'm not saying this
Oh,
experiment
yes. I see what you're saying.
There's actually some very interesting kind of like double-strand breaks happening where they shouldn't be happening. And
Yes.
I can imagine they're very mutagenic in the male germline.
Especially in a species that just recently acquired that in the male germline. Yeah, that's a really good idea. Which species in particular?
Onaceae.
Onaceae, okay, yeah. Well, maybe chat some more, yeah. That's a great idea, thanks. Yeah. Yeah.
So you showed the evidence that in these recombinations mutagenic. Is there other evidence in Drosophila that the combination is mutagenic?
The best evidence that I'm aware of, and once again, there's probably more parsimonious explanations for this, but there is an association with recombination rate in Drosophila and nucleotide diversity. So that's not great evidence. It's usually explained in other ways, but one of the motivations for my study was that, well, in yeast and humans we know what's going on, but how about in Drosophila? So yeah, good question.
Do you know if the mutation has pleiotropic effects on other traits and that the flies might be generally poorly and thus have more mutations?
That's a good question. So we know that the MEIP22 allele that we used does promote non-disjunction in gamete formation. So obviously we selected against, well, only non-disjunction in the sex chromosomes in Drosophila melanogaster is viable. So we wouldn't see any other cases, but we did select against that. Other than that, I'm not aware of any other like well-characterized pleiotropic effects.
Okay, I think we'll have to switch over, but let's thank Rob again.
Thanks, everyone.
Yeah.
Okay, so next up we have Spencer Koury, who's going to be talking about global increases in recombination.
Yeah, absolutely. So this has been absolutely wonderful symposium, and I want to thank you guys both for putting it together. So, yeah, just title slide. This is a really great time to be studying chromosomal inversions. I'm going to tell you about inversions today. Here are just a few of the organisms where their inversions are associated with fascinating range of biological phenomena. Each of these systems has a chromosomal inversion that helps promote adaptation, sex chromosome evolution, species divergence, maintenance of supergene complexes. A lot of really cool stuff. But we also know inversions cause fitness under dominance. They have a really large genetic load, and the vast majority of them go extinct in like less than 20 generations. So like inversions are an absolute disaster for the genome. And this kind of might seem like a paradox, left side, right side here, but the resolution is actually really simple. Inversions aren't intrinsically good or bad. In fact, they only do one thing, and that's just alter meiotic recombination, which is to say that actually, which is to say that both positive and negative effects associated with inversions are just the product of reduced crossing over. And therefore, the key to understanding the evolution of any inversion, or all inversions, is understanding how inversions alter both local and global recombination rates. Okay, so here's what I'm going to do. I'm going to first show you that inversions don't just suppress recombination locally, they also redistribute it globally throughout the genome. Then I'm going to use meta-analysis framework to describe this redistribution of crossing over onto non-homologous chromosomes, and that's what we call the interchromosomal effect, interchromosomal. Finally, I'm going to show you that even though this global change compensates for local suppression on a single generation scale, the importance for evolution is quite small when comparing the deleterious mutation accumulations near inversions. And so I really find this kind of contrast quite interesting because the interchromosomal effect is real, it's true, it causes recombination rate variation. But their magnitude and their diffusion makes them of almost trivial consequence for evolutionary genetics. And so this is kind of like a puzzle that I like. So here's the karyotype of Drosophila melanogaster with the acrocentric X, J-shaped Y, 2 metacentric autosomes, and the dot. Like we just heard, the Y dot and males in general don't recombine, so we're going to ignore them for the rest of the talk. And to remind everybody that crossing over occurs in prophase 1 of meiosis after the genome has been replicated, I'm going to show all the chromosomes in their 4-strand bundle state, just like this. So in absence of an inversion, the recombination landscape looks a lot like this, and this is super smoothed, but it's got a fairly typical mid-arm peak and low recombination near the centromere. This is pretty normal. And when we introduced heterozygous inversions, and we have, that's a IN2RNS, that's a real cosmopolitan inversion. The formation of dicentric chromosomes acts to suppress recombination, or so we hear. So you're probably expecting to see an inversion loop diagram like that, and that really does occur. But if you were to actually do the math, formation of dicentrics explains about 10 to 20% of total recombination suppression. Okay, now that's absolutely crazy, right? Because everybody loves inversions, every textbook has this diagram. But we don't have a mechanistic explanation for 80 to 90% of the phenotypic effect of inversions for the only phenotype that we can be absolutely sure inversions are affecting. So like, my mind was blown, I hope yours is too. So there's several reasons why we know that the formation of dicentrics is the incomplete explanation. First is because we can use compound chromosomes, that's actually taking X's that are on 2 different and attaching their centromeres together. And when we do that, we can recover dicentrics as rings. And when we do that, we see that rings do form, but at rates that are far, far, far below what we would expect given the size of the inversion. Also, recombination is suppressed outside of inversions, and this literally can't be caused by the dicentrics. And the suppression kind of decays with distance as you move away from the inversion. Even more surprising is that the rates of recombination are slightly elevated, or at least they're observed to be elevated on the uninverted arm. Here I'm showing that in blue. And finally, on physically unlinked chromosomes, for some unknown reason, the rates of recombination are also higher, right? So I've just told you a whole bunch of things that might be news, but like things are real weird when you've got an inversion. And this is what I call spooky action at a distance, and it's illustrated in green, and that is the interchromosomal effect on recombination. So I think hopefully that is interesting in and of itself, but now we're going to start kind of digging into what this is all about. Okay, so if you take those 4 effects, and here the arrows are proportional to their effect size on a per base level, it's absolutely dominated by the local suppression. But because they are operating over very different regions of the genome, to a first approximation, these things are about averaging out. So that's actually the redistribution that I'm saying. And I'm not going to say anything too strong about this, but that's the moving to the other chromosomes. So it's almost like there's crossover homeostasis. And I say almost because the necessary experiment of measuring these effects simultaneously for a single inversion on a common genetic background not only doesn't exist in the literature, I mean, it would take me a couple years to do. So, not going to do it. And what I've been showing you is a bunch of different papers, each of which measures one of these effects, and I've put them together in a common model. So if we want to answer the question, how do inversions alter recombination on non-homologous chromosomes in a systematic fashion, we need to develop a mechanistic model, and we need new, better experimental data. Not going to show you that. What I'm going to show you is a meta-analysis. And so what we have is there's actually tons and tons and tons of experiments, and they're from like times when probably everybody in this room wasn't born. And we're going to take all those and we're going to use it in a meta-analysis framework. We've got lots and lots of data, which is great. And then for the mechanistic model, I don't want to harp on this too much right now, but what I'm using is that Foss-Landy-Stahl counting model. Sometimes people call it the gamma model, chi-square model, Poisson-Skip model. All these things are the same family with slightly different assumptions. And the common assumptions are this. Double-strand breaks occur as a homogeneous point process, Poisson point process along the genetic map. Repairs those breaks is a stationary renewal process, and differing assumptions about what sort of renewal that is. And finally, chromosome segregation in the female is a random one-half thinning of those processes. Now, this is probably overload for most people. That's cool. But the really good thing about this is that we can get maximum likelihood estimates of genetic distance, recombination intensity, and crossover interference. And even more importantly, is we can now simulate the sampling distribution for the effect sizes of all those things in a case control standard versus inverted cross. So as long as our meta-analysis data, we get the raw data from those studies, we can essentially calculate anything we want, and I'm quite happy about that. So once we get a good feeling for those effect sizes, I'm going to start talking about SLIM. And that's not going to be a huge part of the project, but I think that's kind of going to be the most interesting part for conclusions. So let's take a look. First thing is, I'm showing you data from about 100 X chromosome experiments. These were all pre-1966, so old stuff. And the good news is there's definitely a recombination intensity increase. And it's higher on the sex chromosomes anytime there's a heterozygous autosomal inversion. Good news. I think we actually heard that result just earlier in the previous talk. In contrast, sometimes you hear this claim that there's extra sensitivity of the centromeric and telomeric intervals to inversion elsewhere in the genome, and that's kind of just a statistical artifact. So when you start doing this in a more common way, there's just no data to support that. And if you take mixed models, actually all those really high estimates up on the top left are coming from one researcher and just a couple of experiments. So like, I really don't think there's a lot of kind of variation along the chromosome. Similarly, there's no real evidence for an alteration of crossover interference. Yeah, coefficients of coincidence change, but in terms of the no effect function, that coincidence function, there's no real change either. So let's focus on this one. And if you're a meta-analysis person, you're probably like, uh-oh, there's big publication bias there. Turns out I don't think so. Because what's happening is each experimenter kind of has their own set of lines, their own set of balancers that they're using. So they're inducing the interchromosomal effect with between 1 and 7 inversions. And once you kind of regress this out, we get a very nice relationship. So we can say that adding a single autosomal inversion causes about a 0.52 centimorgan per megabase increase in recombination intensity on a non-homologous chromosome, specifically the X for this data set. So the meta-analysis supports the primary claim. That is recombination increased on a non-homologous chromosome? Yeah, definitely is. But everything else that we kind of see in the literature, there's not a lot of evidence to support it, right? That doesn't mean it doesn't exist. It's just the evidence is not there in the current state. We've got a kind of estimate of 0.52 centimorgans per megabase increase on the X chromosome. And I will note right now the intra-chromosome on the other arm effect and the inter-chromosomal effect on the third chromosome, another autosome, they tend to be a little bit smaller. So 0.52 is probably the kind of upper level. Now these numbers are still in line with the crossover homeostasis hypothesis. It's like we have a local global balance. And I'm not going to make a final conclusion about this because I need a much, much more precise calculation than kind of the hand-wavy stuff I'm doing right now. But what I'm going to do instead is I'm going to tell you about, even though these green cartoon lines are real, and they're an interesting transmission genetic problem, I'm fairly convinced that they're not important for evolutionary genetics. And the way we're going to explore this is using forward-time population genetic simulations. So I think everybody's familiar with SLIM, and we're building off of a study by Burden et al. That uses SLIM to look at deleterious mutation accumulation inside inversions. So we reparameterized this model to go to match the Drosophila genome, genomic architecture, and we drew out our rates from the data, which is very Scottish influenced over here. And this is the worst slide ever, but it has all the information. This is for people if they want to look at it afterwards online. I'm going to move it now. I hope so. Okay, this is what we saw earlier at the end of the results, and now I'm going to add those evolutionary results in context of our transmission genetic effects. So this is what would happen if there was absolutely no recombination suppression due to inversions. And what we get is a mutation selection recombination balance, which is about 1 deleterious mutant per haploid chromosome arm, and that's uniform across all the arms, excuse me. As soon as we have this kind of naive recombination suppression inside the inversion, mutations are accumulating, and they're accumulating fast. This is on the log scale. There's a small effect outside, which says that even if you have a tiny bit of recombination, it's actually pretty good in terms of efficiency of selection removing deleterious mutants, excuse me. And if we look at the final result, I've actually superimposed those dots over the gray dots. There's really just no effect on deleterious mutation accumulation. I actually have standard error bars, but you can't see them either. So here's what's happening. Mutations are raining down on these chromosomes all the time. Selection is trying to get rid of them as best it can all the time. And if we fiddle with recombination, that might change the efficiency a tiny bit, but it really just doesn't make a difference on non-homologous chromosomes. If we suppress recombination entirely where the inversion is, that makes a huge difference. And so, in terms of conclusion, we can say that absolutely inversions do this thing. They redistribute, but in terms of evolutionary consequences, those diffuse effects really aren't that important. And I think that this is really a great contrast, and I'll end. I've been too long already.
I think we have time for one question.
Yes, ma'am.
Thank you. Yeah, so I'm wondering about actually in lab populations, because it seems you were doing this in like a natural population size, but we who maintain Drosophila in the lab, they've been maintained for decades and decades with balancers. So have you done simulations in like the type of effects of population size that you would have in a typical Drosophila culture, which is a lot smaller?
Yeah, so it's a great question. I'm a lab guy too. This is exactly what I am concerned about. My answer is no, I haven't done it, but of course the effective population size of what's inside your vial is so small that this won't matter either. Right, so it's definitely happening with balancers, it just won't matter.
Okay, I think people have to go to lunch, but I just want to say thank you very much to all the speakers and for all of you for coming. And we'll have our next session in the afternoon. Thanks, everybody.